Inspired Senior Research Fellow. I'm so pleased to welcome you all for my PhD Viva voice. Okay. PhD topic of my research is that bioprospecting of dermatomyces cultures of Goa for understanding bioprocess of submerged pelletization and characterization of melanin as a bioproduct. This research was carried out at Goa University in the Department of Botany under the guidance of Dr.Nandkumar. Coming to the brief introduction, Dermatomyces is a well-known genus which is edible and it is found to be symbiotic associated with termites belonging to subfamily Macrotermitinae. These termites construct a fungus gome underground and they cultivate this Dermatomyces. As you can see in this picture, there is an underground fungus gome which gave rise to the fruit body. And there is a transition phase which gave rise to this fruit body is connected by the pseudorhiza. According to the Index Fungorum, there are around 92 taxa reported and 57 distinct species. This is a complex life cycle how it is in the nature. Termite mounds, when you excavate it and you see that is an underground fungus comb, and on this fungus comb you can see several sporodorsia, which is the anamorphic stage of Dermatomyces. During monsoon, there is a differentiation taking place and their food bodies are produced. They complete their life cycle by discharge of basidiospores, and these basidiospores are taken up by the termites and re-inoculated back into the fresh fungus comb material. This is a distribution of Dermatomyces. You can see it is well distributed in entire African and Asian continent. There are around 44 different countries wherein you find the Dermatomyces species. The highlighted species indicate that there are several taxonomists such as Hein, Pagler, Curran, and Tebua. They have described in the taxonomic description that these species are having a black to grey pigmentations in their fruit bodies. So in India, there are around 27 distinct species. In China, there are 26. In Cameroon, there are 20, and Nepal is 17, which are the highest numbers. So the termitophilic research is mainly focused in many areas, especially like ecological aspect, taxonomist, diversity areas. But we were focused on the bioprocess and bioproduct areas. So this shows a literature review wherein you can see that several bioprocesses related to Dermatomyces have been studied, such as biosorption of metals, which using just chromium metals, saccharification studies, antimicrobial activities, protoplast isolation and regeneration, and protein enzyme secretions. Besides this, the several novel bioproducts have also been reported from Dermatomyces, such as novel cerebrosides, termitosemides, and termitosesphines, which have a role in the Alzheimer's prion disease to cure. So there are several other polysaccharides such as water-soluble, water-insolubles, alkaloids, fatty acids have been also reported from Dermatomyces. Besides this, there are a lot of extensive work has been done on the enzymes. There are several enzymes which are reported, around 15 different types. A recent paper indicate that a sesquiterpene synthase has been reported from this genus. So you can see that this Dermatomyces is highly creative genus with a lot of production of bioproducts which can be used for several industrial purposes. And there is one drawback is that this Dermatomyces cannot exist without the existence of coexistence with termites. In that means in vitro condition, you cannot domesticate to produce the fruit bodies. So it's very much important to get an alternative way how we can produce this biomass to use it for industrial state. So basic understanding of submerged cultures were carried out in this studies. As you've also seen, the taxonomists have described the black to grey pigmentation. That was also taken as a gap in knowledge to lay my aims and objectives. So this is my objectives. First objective, the exploration of wild natural habitats of termitophilic mushrooms for high frequency isolation of pure cultures from diverse species. Second objective, the growth, physiological and cultural studies for selecting and characterizing promising strain for systematic screening. Objective 3, lab-scale production of pelletized biomass in submerged liquid culture and characterization of melanin as bioproduct for selected strains. Coming to the brief materials and methods, the fruit bodies were surveyed in Goa and collected from various natural as well as market sampling was done, and these fruit bodies were processed within 2 to 24 hours. Based on, first of all, they are very specific, habitat specific, there is an easy identification, and the second thing is that it is periodically found only in the monsoon season. Besides this, micro characters were also taken in consideration to identify it after species level by using the standard keys. and various dry herbariums were prepared, spoke prints were taken for the future references. And the pure cultures were done by using standard technique of Stamens and Kilton in 1983. And the colony, 14 termitomyces cultures were subjected and they were grown on Zepecdox medium, and colony compound growth rates were calculated. Bioprocess studies, as you see in the basidiomyces, they produce two major types of morphologies, that is pelletized form or aggregated forms. So based on that an idea, so we had, once we grew the cultures, we had subjected for the various parameters for screening, such as the rheological parameters like culture of cell filtrate was subjected for understand the viscosity, the UV visible spectral study was done to see the metabolites, then the emulsification activities was also carried out. The biomass estimations was done, dry biomass was obtained, and it was subjected to FTR studies for see the chemical composition variation across the species. The morphometric studies and stereomorphological studies were done to distinguish the different types of morphologies across the species, and optical microscopy was carried out along these same studies, and the various images were subjected for biometeorological parameters. So this is the details protocol, but I'm not going to go into detail. Every culture was acclimatized onto the particular medium before going it for the replicate study. Coming to the optimization, various parameters were optimized, and the carbon and nitrogen sources were selected based on the industrially used, and this was done as one variable at a time method, because to see the effect of these parameters on the pellet morphology. Coming to byproduct, melanin was extracted from the fruit bodies because it was naturally found in the termitomyces fruit bodies, and various parts were sorted out like amboresia, stipe, acidorhiza, and was and extracted the UV melanin and subjected for UV visible spectroscopic studies. Besides that, previously the physical characteristics was also understood of melanin by its insolubility test, solubility, bleaching, and so on. The melanization was also seen in the cultures in the solid state, which gave the black brownish pigmentation in the media. They were also selected and screened UV visibly same. And standard technique was used as Saneta 2016 for the obtaining this melanin and purification studies. This is a detailed protocol. And the characterization of termitomyces was done by using various techniques, that is UV visible, optical microscopy, scanning electron microscopy, elemental composition study was done, FTIR, electron pyramidal resonance, XRT, NMR studies, and multi-top studies was done. The inhibitor studies was also carried out using kojic acid and tricyclosol inhibitors, and the zone area was calculated. Results and discussion. Termitomyces fruit bodies, you can see that how they are sold into the market, and this is a very common picture when you see during the monsoon season the roadsides. So each specimen it's cost around 50 rupees as per today's market, and around 25 specimen was costing around 1,200, just 25 specimens. Because of the taste, they have been people are craving for this mushrooms. And this is how it looks in the natural habitats. The pictures, so total 25 field sampling sites were done, and 1,400 specimens were observed, and in 5 market sampling, around 300 specimens were studied. So this is a distribution map of the termitomyces across the Goa. There were only, we found to report around 12 known taxa during the study period. And the common species which was found in the market was Striatus, Mammiformis, then the Albuminosus and Albuminosus species. So this is a descriptive picture wherein you can see how the morphological variations in the fruit bodies. The smallest was reported to be Termitomyces microcarpus, and the largest was reported to be Termitomyces azureus of only 13 centimeter diameter. But in the world, it is known that Termitomyces titanicus is found in Africa, that's the Zambia region. That is the largest, around 2 meters or 1 meter diameter. So the total 19 cultures were obtained during by tissue culturing technique. 6 cultures were found to be growing rapidly, and 13 were slow growing. From this, a 4 millimeter per day cultural growth rate was selected for the further studies of pelletization and melanin studies. So total 8 different species was selected, that is Termitomyces microcarpus, Termitomyces medius, Termitomyces striatus, Termitomyces clypeatus, Termitomyces albuminosus, Termitomyces orienticus, Termitomyces hymi, and Termitomyces globulus. So this is a list of cultures which were used. This picture indicates you how the morphological variation is seen on the Zepedox medium. There were certain cultures which show the banding patterns, like there was dark and light banding of the aerial mycelia, or there were striations which were seen from the center, which was merging out from the center to the radially to the peripheral zone. The certain cultures like the striators and the Clypeators, these were found to be the moderately slow growing, whereas the slow growing and the fast one showed the effuse growth. There was also seen that there was a pigmentations in the Dermatomyces albuminosus, which was produced radially and throughout the colony, whereas in globulus and striators, it was seen that there was a patched production of the melanin or black pigments. So the cultural growth rate indicated that the albuminosus and orienticus showed the leaves which was slow growing, and the highest colony diameter was obtained to be 11.8. Then coming to the pigments, you can see that in striators and globulus you can see the black pigments, and albuminosus is also black pigment. But I told you the difference is that in striators and globulus it was in patches, and it was not that repeatedly you would not get the black pigmentation. Whereas albuminosus it was specific and it was continuously producing the black pigments. Whereas also Clypeator species showed a reddish brown pigmentation in the medium. The optical studies were done to impart and understand the cultural characteristics of this Dermatomyces. It was seen that there were several ring structures, mycelial cords, then the exopolysaccharide bound regions were seen, and specific spiral hyphae, somatogamous hyphae were also seen. So the same studies was done to see the magnified pictures. That was especially we had reported the clamp connection in this species, which was not reported previously. Like Bodha and Acker in 1991 has studied the extensive work on cultural studies using the context tissue and as well as in sporulatial stages. And the Uno et al. In 2018 also performed the cultural studies, but they had used a complex mediums with the malt extract or potato extract along with the synthetic components. But here we have only worked with the not the complex mediums, so that you know what are the different growth factors might be influencing, and the growth will be constant with the same if you use the same nutrients. And these clamp connections were confirmed. According to Dan French in 2000, Dan French Leach, he found that in 2005 he reported that Dermatomyces, it's very difficult to produce a heterokaryon from a homokaryon. So these Dermatomyces do have a heterothallic mating system. So hence we can confirm that these clamp connections indeed confirms that they have a heterothallic nature. And there were also ring structures were seen. These rings indicate that they might have an nematophagous activity, wherein they can trap the various microorganisms and leads to this, which will help them to survival in underground on the fungus growth. Coming to pelletization, this is a picture where you can see the details how the pellets were obtained. This classifies the different types of morphology of the pellets. You can see the flakes, the compact pellets, and the fluffy pellets, and the spikies. So certain cultures produce a single type of pellet morphology, or they produce the mixed forms. So these are the aggregates wherein the loose mycelial aggregates were seen. There was no compaction of the core. So based on these morphological studies, we had given a key to easy distinction and identification of the particular culture what we have studied. So the distinct pellets, if they absent, that means there were aggregates production was taking place. There were Dermatomyces hymes, striators, Clypeators, strains produced these aggregates. Whereas if distinct pellets present, then what type of morphology it is? So if it's spiky pellet, then it would be the albuminosus. If it's fluffy, then it's albuminosus second strain. And the mixed type of pellets were seen by the microcarpus. The compact and flakes were produced. Fluffy or spiky pellets along with it, or compact pellets with spiky. So there are several combinations were possible with these different cultures. So this indicates the quantitative aspects, the dry biomass which was obtained. We've seen that the albuminosus produced 0.72 gram per liter dry biomass and globulus 0.71. When it compared with the number of pellets which are produced, it was seen that the globulus yielded the highest amount of mean pellet biomass range compared to the albuminosus. And the size of the pellets was ranged from the 1 to 12 millimeter, and in the aggregate was seen to 3 to up to 80 millimeters. The various cultural broad characteristics was understood. It was thus seen that the pH changed from the acidic to the alkaline. This might be due to the pellets having the low glucose limitation in the center of the pellets, or it may be possibly the lipopolysaccharide secretion in the culture filtrate. Then the viscosity was also compared. It was seen that globulus, adenomyosis and clypeatus produced the least amount of viscosity when compared with the control. And the emulsification activity was seen. It was very mild emulsification activity. So we can say there is a possibility of biosurfactant in the medium, but I cannot recommend you to be used as industrial level for emulsification, because they are better yielding species which gives you this emulsification. UV visible spectral studies indicated that there are that is absorbance peaks were seen at 230 to 300 nanometers, where it indicated that there is possibility of tryptophan or dopamine intermediates which are released into the culture medium that gives rise to that pigment in the medium, and